because that had pretty much gotten us to a certain point of our lecture. And so the next part, because chapter 4, one of the things that we're looking at in this chapter, we are now looking at first how the cell can make a protein. All right, that's what we've done up to this point. And this, of course, is termed protein synthesis. So if you think about, for example, just the plasma membrane itself, all the different types of proteins that were just in the plasma membrane. That's not including any of those proteins that the cell would have to make as its product. Okay? So protein synthesis itself is very important. The other part that this chapter addresses, what is the life cycle of a cell? Every cell of our body has a particular lifespan. For example, red blood cells, about 120 days. I think we mentioned intestinal cells, about every 24 hours, okay? There are only 2 cells that do not duplicate themselves, and that's going to be muscle and nerve. Now, in looking at these cells, let's kind of put the nerve and the muscle cell off to the side, okay? And let's think about the rest of the cells of the body, okay? All these other cells that do have to make sure they duplicate, all right? In order to do that, there are a few things that have to occur. The one thing they have to do, they have to duplicate their chromosomes. So that means that those 46 chromosomes have to make a copy of themselves. This is called replication. All right? And then we have to make sure that as that cell, one cell becomes 2, that as that process occurs, that half of those chromosomes go to or stay in like the parent cell, and half go to the daughter cell. All right? In order to do that, some processes have to take place. So what we say, okay, is the cell has a life cycle, meaning that that's the time it does its job and it gets done. Whatever happens to it, dies, whatever. But we also have to make sure it duplicates. So, the cell's life cycle, the entire lifespan, it includes phases. Interphase, mitosis, cytokinesis. This is where, if you think about this term, cytoplasm, okay? That is the actual pinching in of the cytoplasm, and we've got 2 new cells, okay? So, interphase. What we're trying to look at here in this cell life cycle, it's trying to say, all right, we've got this cell's got to, number 1, do its job, make its product, okay? And then it's got to duplicate everything in it so that one can become two. During interphase, this is the time the cell is doing its job. All right? So, this means whatever product that cell is supposed to be making, this is the time that it does it. But once the cell moves into mitosis, this process right here is cell division. Okay? This, for example, this is the point at which cells' information, the chemical signals that it's getting, that if it goes wrong, this is when cell division gets out of control and cancer develops. Okay? So, mitosis is strictly cell division. Cell division is not interphase cytokinesis. It is strictly mitosis. Certain things are happening at every phase. Like I was saying, during interphase, this is when the cell is doing its job. It is going to begin to replicate DNA, making a copy. Okay, that's replication. And then carry out normal cell activities. Once mitosis starts, this is what's going to give us 2 new cells, but in order for that to happen, we have to go through prophase, metaphase, anaphase, telophase. These phases tell us something is happening to the chromosomes. All right? At each one of these, not only is it letting us know what is happening to these chromosomes, it is identifiable under a scope. You would actually be able to see the cells, like if you were looking at a grouping of cells, which we'll do, if you're looking at a group of cells, in that group of cells, if you're looking at them, you should be able to point to that one cell and say, oh, that cell is either in interphase, meaning it's normal activities, or based on prophase, metaphase, anaphase. Telophase is a little hard to see with our scopes, okay? But looking at a slide, you would be able to recognize each of these stages based on what's happening with the chromosomes. The final part that has to occur is the cytokinesis, which is literally division of the cytoplasm. Okay? It's where everything else has duplicated, meaning organelles, cytoplasm, cytoskeleton, everything else has duplicated, and we have now pinched off and become 2 new cells. In order for these phases to happen, one thing, very important, the replication of the DNA. Now, DNA, does it leave the nucleus? No. DNA always stays in the nucleus. The replication takes place in the nucleus of the cell. It is simply where we need for those DNA strands to literally pull apart like a zipper, okay, if they do that. We have little proteins and everything that'll help, like DNA helicase and so forth, where those two strands, it almost looks like if we've got that strand of DNA, and everybody knows it's a double helix, right? Okay? It almost looks like at one end there's a zipper. Everybody knows like a little pull thing on the zipper. And it looks like it just simply picks up and just pulls apart. Those two strands open up. All of our little nucleotide bases get to come in and pair up and pair up. And I now have two new strands. It's really cool, okay? That is termed replication, okay? This making of two new strands of DNA. So, when we're looking at it, and I've got my DNA strand, and we all know DNA are those bases A's, T's, G's, and C's, okay? All right? And to match it up, if we're looking at our two strands, A matches to a T, T matches to an A, G to a C, C to a G. All right, is everybody with me? So if it comes time for my DNA to duplicate, to replicate, to make a copy of itself, almost looks like that zipper does this number. These strands kind of open up. Nucleotides get to come in and match up. Does everybody kind of see that? So now, as it unzips all the way up that chromosome, I end up with half of my old strand, my new strand, half of my old strand, my new strand. That means it's semi-conservative because it's keeping half of the what's called original material. So now I end up with two new strands of DNA. Do we want anything to go wrong with our DNA? Oh no, okay? Because DNA, what does it contain? Our genetic information. It contains the genes, and what do the genes code for? Proteins. We don't want that messing up, okay? I don't want the proteins messed up in my plasma membrane. I don't want the proteins messed up that have to stay inside my cell. I don't want the proteins messed up that have to leave the cell. So, this process, now, let's think about this on the scale of how many cells we have in the body. We all hear that term, trillions of cells, right? Okay? Can anybody imagine that number? I would love to have that many zeros in my checking account. Okay, but I don't. All right, so if we think about that number, some of this is occurring continuously. For example, skin cells, as we sit here right now, we're continuously sloughing them off, all right?